Hello everyone, my name is Saranpon Putong. As a previous study about the bison today, I will gonna give you more updating of the data about the bison. And this presentation is already had presented in the EAAP conference in Italy in the 2024. Today is going to be show you on the topic of the relationships of the genetic composition and the carcass grade with the ruminal parameters and the methane emission in the grain-fed bison. First of all of that is our outline today is include the introduction, objectives, material and methods of the study, result and the discussion. The previous study about the life cycle assessment of the bison production that include the cow-calf operation, backgrounding, and the finishing phase. From that preliminary study, we found the biggest part that share on the greenhouse gas emission in the bison production came from the enteric methane. Where is the enteric methane production came from? The bison is categorized as the one of the ruminant. So the bison have a big rumen, and this rumen contain a lot of the microorganisms. One kind of the microorganism we call that is the methanogens. These methanogens produce the methane and emit to the environment. What the factor that involve about the enteric methane production? They can include the genetics, such as the animal size, growth rate, and the microbiome or the normal flora that we found inside the body, and the nutrition type, like the feed intake per day and type and quality of the feed. The bison had a long way evaluation, so nowadays we have the two of the subspecies in the Canada and North America. So that is the bison, or we call that is the plains, and the bison Athabasca that we call is woods. Plain and wood is shown on the different of the phenotype. That the wood had a taller and a bigger size more than the plains, and for the scoring, they have categorized between the subspecies causes that is categorized in the plains score and the wood score. More than that, they try to introduce some of the cattle to have the hybridization effect that can enhance on the production. So nowadays we have to check on the cattle interrogation about the cattle that is mixed cross between the bison. Carcass grading. The grading in the bison is on a different way from the cattle. And this study is aimed only in the A category, that is from ranking the A1 to the A4. And that is calculated on the percent fat deposit inside the carcass. This study have the objective to study on the relationship between the genetic composition, carcass grades, on the effect to the carcass trace, microbial diversity, volatile fatty acid production, and the methane emission. We go to the methods and the method that we use in our study. For the sample collection, we collect all of the 37 bull calves that fed with the barley-based diet, and the age is average less than the 30 months, so that it categorized as a young bison. We collect the animal ID, blood sample, rumen content, and the carcass quality. Blood sample is gonna send the whole blood to analyze by the neogens to do on the subspecies genotyping and including with the cattle interrogation percentage. From our study, the bison genotyping that categorized all of the 32 sample genotyping as the plains, and the score is ranking from the 93 to the 100%. From this study, we categorize into three groups. That is the plain score 100%, and the group number 2 is the plain score is ranking from the 97.5 to the 99%, and the last group is the pain score is less than the 97.5%. For the cattle interrogations, we found the same ranking of the cattle interrogation that is diverse in our study, that is ranking from the 0 to the 3%, so that is also in the same range that the report by the Canadian Bison Association. Carcass trait. We collect all of the grading information, and from our carcass the body weight is average about 500 kilogram, and we collect the information on the live weight, hot weight, grading, and the dew percentage. Rumen samples were collected immediately after they get the sample and snap-feed to transfer to the laboratory. The rumen sample used to analyze the real-time PCR to do on the microbiodiversity, including the bacterial, fungi, protozoa, and the archaea. The methanogen is categorized as the archaea. And moreover that, we analyzed on the gas chromatography to get the word high fatty acid concentration, and the last one calculate back to the methane emission per carcass. The statistic analysis used the glucose values and tests a pairwise comparison. Our result. First, looking about the genetics. The genetics and the carcass trait didn't have any effect on that and didn't show any significant difference between the genetics and the microbiodiversity. For the total word high fatty acid and the acetic acid, they didn't show any difference between the group. But for the propionic acid, they have shown that is higher of the pain score. It shows of the higher level of the propionic acid when comparing group 1, group 2, and the group 3. For the level of the word high fatty acid, we also found that group 1, group 2 is distinct from the group T about the butyric acid concentration, oleic acid concentration, and the carboic acid concentration. The genetics didn't affect any of the methane emission, even if their number is quite different, but they not show on the statistic analysis. We move to another, is on the carcass grade effect. The carcass grade didn't affect any of the carcass trait, including the live weight, hot weight, and the percent yield. And for the total of the microbiodiversity, we saw that is the same of the number that we found in the cattle. And the total word high fatty acid show that it have a trend, a little bit different on the group, but it's not significant different. But when we look inside about each of the word high fatty acid, we found that the two highest group in the grade A1 and A2, it showed different on the acetic acid and the propionic acid when compared to the lowest group. Moreover, almost all of the word high fatty acid is distinct between the group A1, A2 and the group A3, A4, so that the highest of the grading it show more of the production of the word high fatty acid. When we calculate to the methane emission is also following about the word high fatty acid trend, so that is the two highest group it show emit more of the methane more than the two lowest groups. So we found that all of the word high fatty acid can affect on the grading of the carcass and about the involved in the methane emission. Because of the methane production is use the acetic acid as the substrate and turn to the methane and the carbon dioxide emission. And for the propionic acid, that is a substrate to do on the gluconeogenesis, that is support more of the energy for the animal. The level of the butyric acid, oleic acid, isovaleric acid that we found in the highest group of the A1 and A2 is increased more of one of kind that is enhanced on the bond chain volatile fatty acid that involved in the feed protein degradable and increase the microbial protein that is support more of the protein to the animal to create more of the muscle fiber. So in our study, we found a group of the grading A1 and the A2 is have more supply on the protein, energy supply, and they have more of the acetic acid that is support on the methane production. In the conclusion, we found that the genetic competition between the plains and the woods didn't affect any of the microbial composition, carcass trace, or the methane emission. But however, our study only contained the plains group that is in our like a 37 of the animal, but we need more of the sample to analyze more the effect from the genetic composition. On the other hand of the carcass grading, the carcass grading didn't any effect on the carcass trait and the microbial composition. But however, they show that is the grading is different between the concentration of the volatile fatty acid and the methane emission across the grading, especially when we look into the two highest, like A1, A2, compared to the two lowest of the grade A, that is A3, A4. We would like to thank all of our sponsors who support on this research. We cannot break through if we don't have your funding from. For more information and who is interested to do on the research on the bison, you can contact to the Dr. Yuri via the email or the email me. That's all for today. Thank you for joining this. Bye.